The second type of biomolecules that you will learn in this course are your nucleic acids. So we have the DNA or the deoxyribonucleic acid and the RNA or the ribonucleic acid. We know that the DNA is the blueprint of life. All information needed for life are encoded in your DNA, while your RNA, according to the central dogma, are the molecules that relay these information from your DNA to what I call the workers of the cell, your proteins. But aside from these, the monomers of your nucleic acids, called nucleotides, have other functions. For one, you are familiar with ATP, or the adenosine triphosphate, which is known as the energy currency of the cell. If you are familiar with the molecule called cyclic AMP or CAMP, or cyclic adenosine monophosphate, it's a very important signaling molecule inside the cell. Of course, we have the molecule NAD or NAD, the nicotinamide adenine dinucleotide, which is an important cofactor for many enzymes and is an important molecule in cellular respiration. So the function of your nucleotides are not limited to being the monomers of your DNA and RNA. They also have diverse functions. Let's start by looking at the structure of your nucleotides. Again, your nucleotides are the monomers of your nucleic acids. So your nucleotides, each of these molecules have three major parts. The first one is a sugar moiety. The second one is a nitrogenous base or a nitrogen containing compound. And the third one is at least one phosphate group. So again, we have the sugar, which is actually a pentose sugar. So it's made up of 5 carbons, and the sugar used in nucleotides is ribose. So we distinguish between your deoxyribose, the one that we find in DNA, and simply ribose. So the difference is that in the deoxyribose, the hydroxyl group found in the 2' carbon of the sugar is gone. So that's the difference between your ribose and deoxyribose. Your nitrogenous base or nitrogen-containing compound are these. You are familiar with these names, I think. They are adenine, guanine, cytosine, thymine, and uracil. These compounds are classified into 2 major compounds based on the parent structure. So we have the pyrimidine, which is the one containing 1 ring, and we have the purine, a compound containing 2 fused rings. So remember, if it's pyrimidine, the one with the longer name, it only has 1 ring. If it is purine, the one with the shorter name, it has 2 fused rings. So your purines include your adenine and guanine. So you can use the mnemonic PAG. Meanwhile, for your pyrimidines, you have PUCT. or you can add PY. And here we have PU to help you remember them better. Notice that your pyrimidine and your purine has its unique system of numbering the atoms found in the ring. For pyrimidines, it starts with this nitrogen. So this nitrogen is nitrogen 1, carbon 2, nitrogen 3, and so on in a clockwise manner. On the other hand, for your purine, you start with this nitrogen. This is nitrogen 1, carbon 2, nitrogen 3, and so on in a counterclockwise way, followed by 7, 8, 9 on the smaller ring in a clockwise manner. So the attachment of your pyrimidine to the pentose moiety or sugar moiety of your nucleotide is via nitrogen 1. So this is the one attached to 1' carbon of the ribose. So for pyrimidines, it's your N1 attached to 1' carbon of your ribose. On the other hand, for your purine, the attachment is through nitrogen 9. For purines, it's the nitrogen 9 that is attached to 1' carbon of ribose. So the attachment is called N-beta glycosidic bond. It's also a dehydration reaction. Okay, so water is released. Okay, so initially we have a hydrogen here attached to nitrogen 1 of your pyrimidine. So it's the state of the charge of the nitrogen 1 in the physiological pH. So you have a hydrogen attached to it. So it's protonated. So the nitrogen here is positively charged. And then this hydrogen, together with the hydroxyl group of this carbon of the ribose, are removed when this glycosidic bond is formed. So in the same manner, you have one hydrogen from nitrogen 9 of your purine together with the hydroxyl group of 1' carbon in your ribose that are removed as water, forming the glycosidic bond. So again, it's a dehydration reaction. Now your purines, adenine and guanine, both of them can be found in DNA and also in RNA. For your pyrimidines, cytosine is found in both DNA and RNA. However, thymine is a pyrimidine that is almost always found in DNA, while uracil is a pyrimidine that is almost always found in RNA. So take note, there are rare occasions wherein you will find uracil in DNA and you will find thymine in RNA. So the distinguishing feature is the sugar. If it is a ribose, then it is an RNA. If it is a deoxyribose, then it is a DNA. So it doesn't depend on the identity of the pyrimidines nor the purines that you find in the nucleic acid. Now let's go to the sugar part of our nucleotide. So again, the sugar is ribose. And you know that sugars, starting from those with 5 carbons and up, they can form a cyclic conformation. So in the case of ribose, it forms this beta furanose form. So this is your 1' carbon, 2' carbon, 3' carbon, 4' carbon, and 5' carbon. So again, if the hydroxyl group in the 2' carbon attached to the 2' carbon is removed, that is a deoxyribose. Now, for the conformation of the beta furanose, okay, it's not lying on a flat plane. So if you imagine the three-dimensional structure of this beta furanose, all of the 5 atoms that are part of the cyclic chain do not lie in one plane. So two of these, particularly 2' carbon and 3' carbon, go out of the plane. So if we look in this drawing, you have 1', 3', 4', and the oxygen atoms lying in the same plane, but the 2' carbon is out of the plane. So this one can be above the plane or below the plane. If it is above the plane, then we call it the endo conformation because this 2' carbon is on the same side of the plane as the 5' carbon. If the 2' carbon is below the plane, so opposite to the position of the 5' carbon, we call it the exo conformation. Likewise, your 3' carbon can also assume this endo or exo conformation. So there are actually 4 possible conformations for your beta furanose ring. Take note that this interconversion doesn't take place if the beta furanose becomes part of your nucleotide. So if this becomes part of a nucleotide, then we don't expect this to open into the straight chain form. This table shows a summary of the names of our nucleotides based on the 5 nitrogenous bases. So remember, if it is a nucleoside, we don't have the phosphate group attached to the sugar. If it's a nucleotide, then you have at least one phosphate group attached to your sugar. Nucleoside, we only have the sugar plus the nitrogenous base. If it's a nucleotide, you have the phosphate group, at least one, plus the sugar, plus your nitrogenous base. So if the nitrogenous base is adenine, we name the nucleoside adenosine if the sugar is a ribose. So we don't attach any other prefix. If it's adenosine, automatic, you know that the sugar is ribose. If it is deoxyribose, then the name is altered into deoxyadenosine. So you know through this name, you know that the nitrogenous base is adenine and you know that the sugar is deoxyribose. Now, if there is a phosphate group attached to the 5' carbon of the sugar, then we call it adenylate if it is a ribose, or deoxyadenylate if it is deoxyribose. And then we attach, so we place, there's a space, and then if there is one phosphate group, then we say adenylate monophosphate. If there are two phosphate groups, then we say adenylate diphosphate. Or if there are three phosphate groups, we write adenylate triphosphate. However, we are used to using the nucleoside name, right? So we are more familiar with the adenosine triphosphate, cytidine, monophosphate, etc. But this is the more formal, the one that follows the rules or the nomenclature. By the way, I forgot to mention, if the attachment of your nitrogenous base to the sugar is called N-beta glycosidic bond, your phosphate group is attached to your 5' carbon through what we call a phosphate ester bond. So don't forget these terms. So here is a summary of the structures of our nucleotides. So again, we have deoxyadenylate, or the expanded name, deoxyadenosine 5' monophosphate. So this is the one with one phosphate group. Furthermore, we also have symbols commonly used or to represent this nucleotide. So we have simply A, or if we want to remove ambiguity, we attach a small letter D to tell us that this is a deoxyribose. Furthermore, we can expand the name to DAMP so that whoever's reading what we wrote will know that the nucleotide has one phosphate group. Again, if it is a nucleoside, we don't have a phosphate group. So it's the one that is enclosed in the orange box. So without the phosphate group, we would call it deoxyadenosine. So let's have a rundown of the names. We have deoxyadenylate or deoxyadenosine 5' monophosphate, deoxyguanylate or deoxyguanosine 5' monophosphate, deoxythymidylate or deoxythymidine 5' monophosphate, and deoxycytidylate or deoxycytidine 5' monophosphate. So these are our deoxyribonucleotides. For our ribonucleotides, we have adenylate or adenosine 5' monophosphate, guanylate or guanosine 5' monophosphate, uridylate or uridine 5' monophosphate, and cytidylate or cytidine 5' monophosphate. Now, aside from these five nitrogenous bases that we encountered, the adenine, guanine, cytosine, thymidine, and uracil, there are also less common nitrogenous bases found in your nucleic acids. So mainly, these are derivatives of the five nitrogenous bases mentioned earlier. So an example would be 5-methylcytidine. So this is cytidine with a methyl group attached to carbon 5. Another one, we have N6-methyl adenosine. So this time, the methyl group is attached to the amino group that is attached to carbon 6. So notice the difference in the nomenclature. If you see a number, it means this substituent is directly attached to the atom in this position. But if you see an element's symbol with a number next to it as an exponent, then it means that this substituent is not directly attached to the cyclic chain. Rather, it is attached to, in this case, the amino or the nitrogen that is attached to the atom in position 6. So another example, we have N2-methylguanosine. So in this case, we know that the methyl group is not directly attached to the cyclic chain. Rather, we look at position 2 and see that it has an amino group. So the methyl is attached to the nitrogen of the amino group. However, there are some rare nitrogenous bases with their common names, such as inosine. So this purine has a common name, inosine. This one is pseudouridine. It's pseudouridine because it's attached in a different way compared to the normal nitrogenous base. So I mentioned that your pyrimidines are attached via nitrogen 1, which is this nitrogen. But this one is attached via carbon 5 to the ribose sugar. So these are just some of the less common nitrogenous bases that you can find in your nucleic acids.